Once again, hello the future doctors of India to the Study Ayush Forum by the students for the students. Okay, in the previous lecture we have seen the hemostasis part 1, in which we have seen the two important steps in the process of hemostasis. Those were vasoconstriction and platelet plug formation, right? Now today the lecture which I am going to take will be having the most beautiful part of the hemostasis, the very important process of hemostasis, that is coagulation process, the clotting process. Listen it very carefully. There is always a means confusion between clumping and clotting. Clumping and clotting is a very different process. Clotting is a process in which it is done to stop the bleeding, and clumping is done when there is a mismatching. There is an antibody and antigen interaction. So there is a interaction, it is a reaction process. Clotting is not that. It is a different from clumping. So always remember there is a difference between clumping and clotting. Those are very different terms. Okay. Now let us start our today's topic, that is hemostasis part 2. Third and very important, till now we have seen that there is a vasoconstriction, right? Vasoconstriction due to secretion of serotonin, that is 5-HT. It is a local phenomenon, right? Second process we have seen platelet plug formation, the adherence of platelets, aggregation of platelets by protein by platelet activating factors. Platelet secretes thromboxane A2, and also one more important chemical, that is ADP, that is adenosine diphosphate, that we have all seen. If you want that, check our previous lecture, it is detailed studied in that portion. Now when, if this is a blood vessel, right, and here there is a somewhere is the injury, and here the platelets came, and we have seen that here the plug is formed till now, but this plug is very temporary and very loose, so it can break. So what happened here, here comes the fibrin threads, and they tie it, they make the knot, and it makes them stable and now thick clot and very strong clot. So what, how, when this process runs, this is a very cascade process, very much long process. Now what is the mean of cascade? I am saying from last lecture we are studying from 11 12 from the junior classes. What is the mean of cascade? Cascade is a meaning is that the one step is here, when this step will complete, this step will start. If this step doesn't complete, this step will not start, and till next the step they will go successively till the end result is obtained. All this called as cascade process. Clear? This was all about cascade process. Now let us see of coagulation. Now let us see coagulation of blood. What is the definition? Very important definition. Coagulation or clotting is defined as the process in which blood loses its fluidity and becomes a jelly-like mass few minutes after it is shed out or collected in a container. Okay. Now we have seen when we take the blood in a laboratory, it makes a clot. It makes in vitro also and in vivo also, in our body also and in outside also, right? Now it loses its fluidity. Ability to flow is termed as fluidity and becomes a jelly-like substance few minutes after. Approximately 2 to 3 minutes is your bleeding time, and max to max 5 to 9 minutes is your clotting time. Okay, it's shed out and collected in a container. Now how this all clotting is done, what is the mechanism behind it, and what are the three there are 13 factors, not 3, there are 13 factors involved in this cascade process, right? Now how all this happens, let us see in detail. Coagulation of blood occurs through a series of reactions due to the activation of a group of substance necessary for clotting are called as clotting factors. These are 13 clotting factors. They were named according to their inventory scientist. Only the I think so only the ninth one is not according to its scientist name, it is given according to its patient name. Okay, but all the others are given by their scientist names. So how to remember it? There are many tricks available on social media, available on Google, so you can learn. Now I have made the trick in Marathi language. It is very weird, but it's I have learned like that. So I am a creative person, so I like to make more and more mnemonics and all. So it is in Marathi language. So if you like, you can learn it like that, or else you can make your own. No problem. But ultimately, you have to learn it on your. You have to learn on your own, and this should be very important because if you make your memory sharp, you keep the all concepts clear, you will become a good physician. Now let us write. See, फक्त प्रेम तुझ्यावर केअर लावून fifth factor is sixth factor, presence of sixth factor is not proven, so गायब झालीस. सुरुवात आता क्रिसमसला सुरुवात आता क्रिसमसला शांतीत परत हॅपीली फॉरएवर करो. This is a statement which I have made. फक्त प्रेम तुझ्यावर केअर लावून गायब झालीस. सुरुवात आता क्रिसमसला शांतीत परत हॅपीली फॉरएव्हर करू. Factor 1 is your fibrinogen. Factor 2 is your prothrombin. Factor 3 is your thromboplastin. Factor 4 is your calcium. Factor 5 is your labile factor, or it is also called as proaxillerin or accelerator globulin. Factor 6 presence is not yet been proved, but many scientists believe that it is present. Factor 7, it is a stable factor. Factor 8 is an anti-hemophilic factor, which you have learned in our elder classes in your this genetics chapter. Anti-hemophilia is a disease, right? It is a genetic disorder. Factor 9 is a Christmas factor. Factor 10 is a Stewart-Prower factor. Factor 11 is a plasma thromboplastin antecedent factor. And factor 12 is your Hangeman factor. And factor 13 is your fibrin stabilizing factor. All these factors are very important in your blood clotting coagulation process. Right? Now let us see next. Now there are 3 stages in blood clotting. Usually this is a final step, and this is the second step, and this is the first step, right? Conversion of fibrinogen to fibrin. In class till class 12 we have studied that here there is a prothrombin which converts with the enzyme thrombokinase to thrombin. Now this thrombin converts fibrinogen to fibrin. right? This is all the process we have studied. Now, the very first, this prothrombin is present in inactive format in our blood. So, all the which cascade process what we are talking about is to activate this prothrombin. This prothrombin is a king. It requires a very long steps to activate this prothrombin. There are usually two pathways in which this prothrombin is activated. First one is intrinsic pathway and second one is extrinsic pathway. Now what is the intrinsic pathway? This pathway is initiated within the blood with the help of platelets. Via platelets it is initiated, so it is called as intrinsic. And extrinsic is the pathway in which the tissue are involved. Injured tissue secretes thromboplastin, and with the help of that further the process goes on and prothrombin is activated, and then prothrombin is converted into thrombin with the enzyme thrombokinase, and this thrombin converts fibrinogen to fibrin, and this fibrin polymerizes and make a thick clot by making cross bridges and all, and a stable blood clot is formed. Now let us see how the prothrombin is activated. So, intrinsic pathway for the formation of prothrombin activator. In this pathway, the formation of prothrombin activator is initiated by platelets. As I have told you, within the blood itself, it is in the blood only, so it is called as platelets, which are with blood itself. Now, what happens is when you get injured, your endothelium gets damaged and collagen is exposed. Now, when this collagen is exposed, what happens is in blood, inactive factor 12 called as Hageman factor. Okay, inactive factor INC, inactive factor 12 with the help of enzyme callicrine and high molecular weight kinogen converted into active factor 12. Now, what happens is this active factor 12, this is Hageman factor, right? Hageman, Hageman factor. Now, this active factor 12 plus inactive factor 11 factor name plasma thromboplastin antecedent, right? This inactive factor 12 in presence of this same enzyme high molecular weight kinogen converts into active factor 11. Now what does 11 do? 11 do partiality. It does not go to the 10th. He is not loyal, right? So what this active factor 11 does is, this active factor 11 plus it converts inactive factor 9 to active factor 9. In presence of factor 4, which is your calcium. Now what this active factor 9 this active factor 9 is very loyal. This active factor 9, what it does is, active factor 9 plus inactive factor 10 converts into active factor 10. This active factor 10, how it converts? With the help of factor 4 and factor 8, which is anti-hemophilic factor, right? Now what it does is, this factor 8, it converts factor 10 in active format. Now what happens is, this factor 10, active factor 10 plus platelet platelets, phospholipids. Platelets, phospholipids present on surface of platelets. What is done is plus active factor 5. Now you will ask question how this factor 5 activated. It will come in the next step. I will clear it you in the next slide. Okay, this active 5 this factor 5 is present in the blood in inactive format, but it is activated by a positive feedback mechanism in a step in which thrombin is formed. Initially formed thrombin, what does it? It's activate more and more amount of factor 5, okay, by with the mechanism of positive feedback mechanism, which we learn in homeostasis, right? What it does is this all together activates prothrombin. Now this was intrinsic pathway within the platelets, right? Now in this our prothrombin is activated. Now extrinsic pathway, it's a very simple pathway rather than intrinsic pathway, but extrinsic pathway usually occurs more because we get injury to tissues. I'm not saying that blood vessels are not getting injured, but most of all like little injuries are getting on tissues, right? Now extrinsic pathway or the formation of prothrombin activator. In this pathway, the formation of prothrombin activator is initiated by the tissue thromboplastin, which is formed from the injured tissues. Thromboplastin, we have seen it is a factor, it is third factor, right? So this third factor is secreted by injured tissue. What this third factor has is it has three components, which are proteins, glycoproteins and phospholipids. Now the glycoproteins and phospholipid, what they do is glycoproteins and phospholipids, now phospholipids tissue phospholipids, right? Tissue phospholipid in presence of factor 4 and 7 converts, means activates factor 10. Now again this activated factor 10 and plus activated factor 5 plus platelet phospholipids activates prothrombin. In this way, that there are two processes which are called as extrinsic pathway and intrinsic pathway, and in these two ways prothrombin is activated. And now what this activated prothrombin do is, next step we will see, this prothrombin converts into thrombin. In presence of enzyme thrombokinase. With the help of calcium, right? Now this thrombin is very essential. When this is formed, surely there will be a blood clot. For testing purpose, if you have seen that the formation of thrombin is seen, then surely you can say that there is a blood clot. Blood clotting is all about thrombin formation. Once the thrombin is formed, it will definitely leads to the blood clot formation. Now what happens is, when this thrombin is formed, it makes more and more thrombin. But the initially formed thrombin activates factor 5, fifth factor, which is very important, and it activates it via positive feedback mechanism. I have told you earlier. Now what this thrombin does is, it converts fibrinogen fibrinogen, fibrinogen to fibrin threads with the help of enzyme thrombin, right? These fibrin threads are formed, and this thread goes and tie over the knot to this blood vessel or these platelets and make them stable. Initially, these fibrin threads are thin, but later on it becomes dense with the and it becomes stable with the last factor is called as stabilizing factor, factor number 13. With the help of that factor, it becomes stable. Now, it is said that how fibrinogen is converted into fibrin. Fibrin is nothing but activated fibrinogen. The fibrinogen, see, it looks like this, not a perfect diagram. I am only giving this for your understanding purpose. If we call this as fibrinogen, okay. Now, when in each fibrinogen molecule, if two fibrin molecules separates, two fibrin molecules removes, and the remaining this is called as fibrins, and this fibrin further polymerizes and forms the fibrin thread, and this thread ties over that platelets and your clot is formed, and this clot becomes stable thick with the help of 13th factor present in your blood. It's called as factor 13, stabilizing factor, right? Now you can see I have brought the yesterday's diagram itself. This is yesterday's diagram. So here you can see platelets are coming, right? Primary plug is formed. This is collagen. Now here there is a fibrin threads are coming and becoming the dense thick clot and stable blood clot is seen, right? It's a very nice diagram to understand. See, it is an endothelium, it get ruptured, etc., right? It's from cross bridging, cross linking, fibrin cross linking formation, and a stable blood clot is formed. So for today and for this topic, thank you. Be studying, be a good physician, keep your doubt clear and continuously ask question and keep reading the books. The more you read, the more you understand. The more your doubts are clear, the more nice doctor you will be. Thank you. Till that, study well. Thank you.